When we talk about bioinformatics, computational biology, and systems biology, we are really describing three closely related but distinct areas where biology meets computing. They overlap to a great extent, and together they form the foundation of modern biological data science. Bioinformatics focuses on data, that is, developing and using computational tools to store, organize, and analyze biological information. Whenever we deal with DNA sequences, protein structures, gene expression data, or biological databases, that is bioinformatics in action. It is the toolbox of the modern biologist, that is, handling large datasets, automating repetitive analyses, and ensuring results are reproducible. Computational biology, on the other hand, is more about understanding and modeling. It uses algorithms, statistics, and simulations to explain biological behavior. For example, modeling how mutations affect protein structure or how populations evolve over time. Computational biology often builds on the data processed by bioinformatics to generate new hypotheses and mechanistic insights. Then there is systems biology, which takes a step further and looks at biological systems as a whole. Instead of studying a single gene or protein, systems biology explores how many components interact, that is, genes, proteins, metabolites, and pathways, in order to produce complex behaviors in cells and organisms. It often combines large datasets with computational models to understand networks and feedback systems, such as gene regulation or metabolic flux. You can think of it this way. Bioinformatics is about collecting and handling biological data. Computational biology is about analyzing and modeling that data. Systems biology is about integrating many types of data to understand whole systems. Modern biology is truly a data science. Over the last 20 years, new technologies like DNA sequencing, RNA-seq, and mass spectrometry have revolutionized how we study life. Every experiment now produces enormous datasets, far too large to analyze by hand. Take genomics as an example. When the Human Genome Project was completed in 2003, it took over a decade and billions of dollars to sequence a single human genome. Today, the same task can be done in a few hours for under $100. And we are not just sequencing humans, we are sequencing bacteria, viruses, plants, and entire ecosystems. The result is a flood of biological data, such as sequences stored in databases like GenBank, protein structures in the Protein Data Bank, expression datasets in the Gene Expression Omnibus. This growth has changed biology from a largely experimental discipline into one that is both experimental and computational. However, this also brings challenges, as the data are large, messy, and diverse. To make sense of them, biologists need to automate analysis, ensure reproducibility, and integrate information from multiple sources. And that is exactly where programming and bioinformatics come in, as they allow us to turn raw data into biological insight efficiently and reproducibly. This picture illustrates an example of a common bioinformatics problem, which is analyzing sequencing data. In the laboratory, mRNA molecules are extracted from the cells in a sample. Our genes reside as limited stretches on the chromosome. When expressed, they are transcribed into an mRNA molecule. The mRNA molecule is thereafter translated into a protein. The number of mRNA molecules transcribed for a gene commonly represents a gene's expression level, so that the more mRNA transcribed, the higher the expression level. Proteins are generally highly unstable and difficult to sequence, but the mRNA molecule is commonly a stable molecule and easier to sequence. Hence, messenger RNAs have been the target of sequencing. The sequencing produces sequences in FASTA format, usually stored in a plain text file. This is an example of sequence in FASTA file format. We will now take a look at its different parts. Each sequence starts with a header line, see arrow 1, and this header line always starts with a larger than sign, see arrow 2. Hence, the larger than sign gives that here starts a new sequence. Beneath the header line comes the actual sequence, see arrow 3. Each sequence also always has an identification number, ID, which is a combination of numbers and English letters, see arrow 4. The header line commonly also includes some other information, such as the name of the sequence, some information of its function, and so on. This is a common problem in bioinformatics. mRNA sequences are translated into protein sequences by translating a codon of 3 nucleotides into one amino acid. Moreover, the codon ATG specifies where the translation should start. This start codon is often located within the beginning of the sequence and not at the very start of it. And an mRNA sequence can actually contain more than one start codon. That is, the ATG triplet can occur at several places in the sequence. The bioinformatical problem will be to find all occurrences of ATG and report the codon's start position, which is given by the position of A. This will give a list of all possible starts for translating the mRNA sequence into an amino acid sequence. We will now talk about problem solving a bit, since this is a very important step in bioinformatics. Problem solving is to find a solution to a described problem. A problem could be calculate the sum of two numbers, pass a text file, and output some requested information, or analyze FASTA sequences to find the number of A's in the sequence. We can divide problems into two different types, ill-defined and well-defined. If we have a problem that is ill-defined, it will be difficult to implement it so that the program solves the problem. A well-defined problem is much easier to implement. An ill-defined problem has no clear goals, no description on how it should be solved, nor any expected results. A well-defined problem has specified goals, a clearly described solution, and expected results. Important to realize is that shit data commonly means shit results out. If you have an ill-defined problem, you will get an ill-implemented program. Therefore, spend some time to write problems that include clear goals, a specified solution, a path to implementing the solution, and an expected result. It will be of benefit for you as a programmer. Here are two examples of a described problem. The first one illustrates an ill-defined problem. It is short with no details about expected input or output, nor any hints about how the problem should be implemented. The second description of the problem is much longer and contains more information that is needed for doing a good implementation. It gives us what the expected input is, the expected output, and the purpose of the program. Of course, one needs to know what a protein motif is. It is a sequence pattern of amino acids in a protein. Certain regions or specific amino acids are more conserved than others among closely related proteins, that is, proteins that carries out the same or similar function. Such regions are often conserved because they encode a part of the protein that is functionally important. There are many motifs that are conserved among different proteins. These motifs can be described as a pattern, also known as a regular expression. From problem description 2, we can extract important information for our solution path. First, we can identify what should be the input to the program, that is, a protein sequence and the motif. The output should be a message to the user informing if a match has been found or not, which indicates that the motif is present in the sequence or not. Thereafter, we can break the problem into several sub-problems, which are smaller, more detailed step of the problem. This will facilitate the implementation of the problem and making the program. The first thing is to store the protein sequence as a string in the program. This is a common programming task. In order to process the sequence, it needs to be stored as a string within the program. Second, the motif must be converted to a so-called regular expression, which is also a programming task. In order for the program to identify if the motif is present or not, the motif has to be represented as a regular expression within the program. Third, the regular expression is used for doing a match against the protein string. If the regular expression is present in the string, then it matches. Otherwise not. Fourth, we construct two messages, one for if a match is found and one for if a match is not found. Depending on the result of the matching, one of the messages will be printed out. In bioinformatics, we often start by working with raw data files. These files contain biological information in standardized text-based formats, which allow researchers and tools to communicate effectively. For DNA or protein sequences, we often use the FASTA format, which we covered earlier in this video. Each entry begins with a line starting with a greater than symbol and an identifier, followed by the sequence itself. When sequencing reads are stored with quality scores, we use the FASTQ format. For gene annotation data, which describe where genes and exons are located in a genome, we use formats like GFF or GTF. These files are like maps of the genome. Structural data are stored in PDB files, which record the 3D coordinates of atoms in a protein. And expression data, like RNA sequence counts, often come in spreadsheet-like CSV or TSV formats. Each of these file types serves a specific role in the bioinformatics workflow. Learning how to read and process them programmatically will be one of the main practical skills for a bioinformatician. So, what do bioinformaticians actually do with all this data? Their tasks span a wide range of analyses, where some are quite simple and others extremely complex. At the most basic level, you might analyze DNA sequences. For example, calculate GC content, find open reading frames, or search for motifs like promoters or restriction sites. You might align sequences to see how similar they are, or search databases to find related genes using tools like BLAST. In other projects, you could analyze gene expression data to find which genes are active in certain conditions, or explore protein structures to study folding and function. Bioinformatics also includes data visualization, such as heat maps and genome browsers, and workflow automation, where scripts and pipelines handle hundreds of samples automatically. A large area is also development of algorithms and tools.